Hello everyone, this is Dr. Pooja Johari, your biochemistry mentor. Now today we are going to discuss a very important topic. In this video, we would be discussing about the transport of glucose. Now if you see my previous videos, they were on chemistry of carbohydrates because this is how we are going to plan. This is how we have to prepare. So carbohydrates mein first we studied the chemistry. Now if we come over to the metabolism part, because actual biochemistry starts when this glucose, galactose, whatever classification we studied in chemistry, how they enter inside the cell, what happens to them when they enter inside the cell, this all we are going to cover. Now to start up with the basic, I just want to tell you one thing. We are eating the polysaccharides, isn't it? Jaise bread we are eating, rotis we are eating, puris, whatever carbohydrates we are eating. The moment we start eating these polysaccharides, they will undergo stepwise degradation. It is not like that ki the moment we eat and it will be immediately converted to glucose. No, slowly it will be converted. And the digestion of carbohydrates always starts with the salivary amylase. We know that. Okay. Or you can say amylase is the first enzyme which is acting. The moment it enters and finally when it reaches the intestines, they are completely degraded to form these monosaccharides. Now the question is, once the polysaccharides are degraded to form monosaccharides, what happens to these monosaccharides? So the principal monosaccharides, if we talk about, they are basically three. And what are they? Obviously, the one which is the hero, that is glucose, followed by galactose, which is the component of the milk sugar, and fructose. And these monosaccharides will now undergo absorption. And after absorption, they will undergo assimilation. So this is where the actual biochemistry starts. This is simply digestion and absorption of the carbs, and this is actual biochemistry. Now the thing is, whether it is glucose, whether it is galactose, whether it is fructose, how they are carried to the respective organs and they are stored. Because always remember, liver is always the kitchen of the body. Just during the time of COVID, we used to store everything in the storehouse or in the godowns. Same is the case with liver. Whatever is there, it will be stored in the liver so that when the body is undergoing fasting from the liver, they will be sent. Because again, keep one thing in mind, just liver is the kitchen of the body, heart and brain are the two organs of our body which just cannot stay without energy for even a parsec of a second. So they would be always requiring energy, right? Now the question is how they undergo absorption. Ab absorption ke liye obviously transporters will be there. It's not like that ki glucose will just walk away from the intestine to the liver. Okay, or it will just walk away towards the kidneys. So absorption ke liye we require the transporters and we refer them to as glucose transporters. Very, very important. So in this video, I would be discussing the glucose transporters in detail. And commonly they are known as GLUTs, where GLU stands for glucose and T stands for obviously transporters. Now coming over to this, we have already studied different ways of transport in physiology. There are, overall, if I have to just highlight the things, I can simply say that they can be active, it can be passive, right? We know that the basic difference is active energy is required, passive mein no energy is required. It is along the concentration gradient. Okay. Then coport is there, symport is there, antiport is there, and uniport is there. So they are different type of transport mechanisms. So here what we are going to discuss is these glucose transporters are first of all categorized into two classes. One is sodium dependent. And since they are dependent on sodium, they are of two types. Obviously, S will be added as a suffix. So here S stands for sodium. So S GLUT 1 and S GLUT 2. So one class sodium dependent, the other is sodium independent. Now, the question is, what is the difference between sodium dependent and sodium independent. So if we talk about the basic differences, because this transport of glucose mechanism we have to understand, because this is very, very important. Physiologically, biochemically, it is important. Most of the drugs, hypoglycemic drugs, they act on this mechanism only. So if we talk about the first basic differences, sodium dependent is actually known as, it is actually active transport. Sodium independent is actually passive. Right. So if we talk about active, so we all know it is always against the concentration gradient. So since it is active, it is against the concentration gradient. And if we talk about sodium independent, it is along the concentration gradient. So we can simply call it as diffusion also, which we know that. But here diffusion will be carrier mediated. Matlab no energy is required, but yes, carrier will be there and facilitative mode of transport will be there, which we will be discussing. Here I am just giving you the, I would say, the broader way of differentiating. So I can say one is active, other is passive, against the concentration, along the concentration gradient. Now if it is active, it is against the concentration gradient, requires energy. Along the concentration gradient does not require energy. Only it is carrier mediated, carrier required, but no energy is required. Simply it will just flow like that, diffusion. Then fourth important point here is that this is unidirectional, very, very important, unidirectional. It will go only in one direction. Because body cannot afford whatever is going along, which is requiring energy, body cannot, afford every time to expense that much energy. And if we talk about this, it is bidirectional. Very, very important. And it follows a ping-pong mechanism. Ping-pong mechanism is just like a, a match of table tennis. Just say table tennis whenever we play. So it is like between two bats and the ball. So wherever the concentration is more, it will just flip over there. It will empty whatever glucose is there. It will come back and again it will strike. So it is ping-pong mechanism. Or commonly we can call it as simple diffusion also. And it is facilitative mode. Facilitated diffusion. What is facilitated diffusion? Obviously, it is carrier mediated, but no energy is required. So I can say these are the four important differences. Now here one more thing I just want to write, that since here glucose is the one which is helping glucose for its transport, so we can simply say that it is also known as secondary co-transport. I'll explain the meaning with the diagram. So it is also known as secondary co-transport. So this is basically, you can say, two type of transportation: sodium dependent, which is actually active and which is also known as secondary co-transport, and it is unidirectional. On the contrary, sodium independent is passive, it is bidirectional, and it is simply facilitated diffusion. Now we are going to discuss these transporters one by one in detail, because clinically also this is very important, theoretically it is very important. In fact, this is the most important topic. Why? Because it is with the help of transporters only the cells can have glucose, the cells can have fructose. And always keep one thing in mind, these carbohydrates are the universal energy carriers. If we can restrict carbohydrate diet, we can actually reduce the intake of glucose, and it can actually help in weight loss also, if we see biochemically. Okay, so first of all, let's start up with sodium dependent. So I would write this A, sodium dependent transport. Right. Now, just now I mentioned it is unidirectional. Okay, it can occur in one direction only. So if I have to explain with the help of a diagram, say this is the lumen and say this is the blood. Okay. Now this is carrier protein, right? And the same carrier protein like this. Now what is happening? You can see that this carrier protein has two binding sites, 1 and 2. So what happens here is, in the first binding site glucose and in the second binding site sodium is going to bind. Matlab, this carrier protein obviously will be rotated only when it is having both. Means glucose, in a nutshell, in a layman's language, I would say that sodium is a prerequisite or sodium is a mandate to attach itself with a binding protein, then only glucose will come in mind. But then it's simple. It will be rotated at a 360-degree angle. What we require is, we require glucose. So glucose will enter into circulation. Regarding sodium, sodium has to pump back. Now sodium will be pumped back. Why sodium will be pumped back? Because sodium has to again bind with more of glucose. Now here the most interesting point which you have to understand is, glucose will always move along the concentration gradient, always. But sodium is the one which is moving against the concentration gradient, because if you talk about sodium potassium ATPase pump. Okay, so with the help of sodium potassium pump, this sodium will be pumped out. So actually sodium is against the concentration gradient, but glucose is along the concentration gradient. And obviously this is going to happen, no, because we are having a diet rich in glucose. So glucose ka concentration automatically would be more and it has to be sent in circulation. So glucose will move along the concentration gradient. And since energy is required but it is indirectly for sodium, that is why it is, or I would say, indirect use of ATP, of energy in the form of ATP, because we know that ATP is the energy currency of the body. So indirect use of ATP leads to secondary co-transport. Very, very important. I am not going into much detail because again this you are going to study in detail in physiology also. So here you just have to remember that sodium is the mineral which is required for the transportation of glucose from the luminal site of the intestine into circulation. This is present in the kidneys also, because again there also will be absorption and reabsorption of glucose, because intestine and kidneys are the two main organs which play a very, very important role in the metabolism of glucose along with liver, because liver is basically acting as a storage site. Okay, so this is sodium dependent. Quickly you can say that from the luminal side it will be rotated and it is going to give glucose into circulation, and automatically glucose will be sent like that to the cell. Now the question is, since I mentioned 3 monosaccharides, glucose I mentioned, galactose I mentioned, fructose I mentioned. So glucose and galactose are with the help of sodium. So I can write glucose and galactose will require sodium dependent, whereas fructose has altogether a different glucose transporter which we will be discussing. Fructose is transported by GLUT5. Okay, so sodium ke liye, sodium requires glucose and galactose, or vice versa, glucose and galactose require sodium. Now since I told you they are 2, so I can say that one is S-GLUT1 and second is S-GLUT2. Now the question is that where they are present and what is their function. So if we talk about S-GLUT1, S-GLUT1 is present in the luminal cells of intestine, or you can simply say lumen of intestine, and it is present in the PCT. Similarly, SGLT2 is present in the proximal convoluted tubules of the kidneys. Now, coming over to this, GLUT1 is required for both glucose and galactose, and GLUT2 is only required for glucose. And the basic difference between these two is that GLUT1 is for absorption of glucose and obviously galactose, whereas GLUT2 is for reabsorption of glucose. So I can say that these are the two main glucose transporters dependent on insulin. Right? So simply if you see, if I highlight it, GLUT1, absorption of glucose and galactose, and it is very interesting to know that if we compare the absorption rate of glucose and galactose, galactose is 100 times more easily absorbed than glucose, and GLUT2 for the reabsorption of glucose. Now what is the clinical application of this? Very important again. If we talk about the clinical application in renal glycosuria, there is mutation in the SLC55 gene. So it leads to decreased renal threshold, right, thereby increasing or leading to glycosuria. And it is clear from the word itself if there is a mutation. So obviously what will happen? Renal threshold of kidneys will decrease. So it will lead to glycosuria, which is one of the important reasons of this. Second important point which you have to remember is that whenever ORS is given, because whenever there is excessive dehydration, you can say in vomitings, in diarrhea, ORS is given. So what is that ORS? Always remember it is nothing but it is the combination of salt and sugar. So why salt and sugar? Because sodium and glucose are absorbed together. That is why sometimes we make ORS at home also. So what is advised? We have to mix equal volumes of equal volumes of sugar, equal volumes of salt. The reason behind this is that sodium is the one which is responsible for the transport of glucose. Okay. And third important point here is very, very important. What is the pharmacological association? Always remember glyphosines. What is the pharma correlation here? The glyphosines are basically GLUT2 inhibitors. They are the inhibitors of GLUT2. And GLUT2 is responsible, if we talk about this, I can simply say that, right, and this is how it is going like this. So what is happening is that GLUT2 is responsible for the reabsorption of glucose, and this is your SGLT2. It is reabsorbing glucose. But if this glyphosines are inhibiting it, this dapagliflozin is there, it is mechanism of action is glyphosine when it is given to a patient suffering from type 2 diabetes mellitus. So what will happen? It will act as an inhibitor. So obviously it will lead to increased excretion of glucose through urine. Right. So because in diabetes what is happening is the concentration of glucose is already too way high in the cells or in the blood. So the mode of action of these hypoglycemic drugs is they have to bring the level of glucose down. So what they will do is they will increase the excretion of glucose. And if they increase the excretion of glucose, glucose will be excreted in urine. The only drawback of use of glyphosines is chances of UTI. Why? Because again, In urinary tract infections, what will happen is since the concentration of glucose is coming out, glucose is there in the urine, so it will give rise to, it will be like a, kind of home for the bacteria. The only disadvantage of gliflozins is this. But again, it can be corrected by maintaining proper hygiene. Okay, so this is a very important mode of action of these gliflozins. So I can say gliflozins are GLUT2 inhibitors, and that too SGLT2. Now don't mix GLUT2 with SGLT2. There is a difference. This is sodium dependent. Okay, and we can simply say that here it will lead to glycosuria. The only disadvantage is UTI, but it can be easily managed. That is not a very big issue. So this is what these are the different type of sodium dependent glucose transporters, where you can simply see it is unidirectional. There is no bidirectional one. It cannot come back, and it is also known as secondary co-transport. Then after this, then we come over to the other glucose transporters, which are sodium independent. So if we talk about sodium independent, we will simply call them as GLUTs. Now they belong to a category again, no sodium. So they are again of two types. One is insulin independent and second will be insulin dependent. Insulin independent and insulin dependent. And if we talk about the characteristics of this, simply you can say that it is facilitative diffusion. It is along the concentration gradient, very important. Right? And it is bidirectional. And fourth important point is it follows ping pong mechanism. Now, if we talk about the graph, what kind of graph is obtained in this? So I can simply say that here we can take the solute concentration, and here we will take the rate of glucose transport. So if we talk about simple diffusion will be like this. Obviously, simple diffusion along the concentration gradient, more the concentration, more will be the transport. But when we talk about the facilitative mode, it is like this, little bit like this, or I would just make it into a more straight line. It's actually straight line. So I can just make it like this. So this is simple diffusion and this is facilitated diffusion. Facilitated diffusion. Just like saturation kinetics. Okay, so this is simply, kind of graph which can differentiate between simple and facilitated diffusion, and this is how glucose is transported. And if we talk about ping pong mechanism is simple. Just is suppose here it is having glucose, right? What will happen? This will rotate. It will empty the contents like this. And this is how glucose will be released in circulation. This is ping pong. Now the question is, first of all, ये तो हो गया कि how they are transported. Now what are the different type of GLUTs? What is their significance and where they are transporting this glucose? Because every organ has a specific glucose transporter. So first of all, we are going to list, just again make a table. Now if we talk about and like this. Now this here I can say that this is the name of the transporter. Second is where they are located and what is their function. So if we talk about, let's start with GLUT1. Now if we talk about GLUT1, always keep one thing in mind, that GLUT1 is the most specific transporter of glucose in the RBCs. The most important role of this. And always remember that abhi we will be discussing the different stages in the body, because always we know that all the time we cannot eat, no? So we are having proper meal. Suppose we are having breakfast, we are having lunch, we are having dinner. In between, if we are not having anything, if I talk about the concept of intermittent fasting, what we are doing is we are telling our body to take some rest. Okay, so that is what is the fasting stage. Whether it is early fasting, whether it is late fasting, whether it is prolonged fasting, whether it is prolonged starvation. But always keep one thing in mind, no matter what is the status of the body, even if we are not eating for consequently 4-5 days, RBCs will always take glucose for energy, because poor chap RBCs, they lack mitochondria. So their ultimate source of energy has to be glucose via glycolysis. Okay, so ये जो GLUT1 है, this is specifically for RBCs. It is for others also. For the brain also it is required. For the kidneys also it is required. For the colon it is required. And for the placenta also it is required. And the most important function of GLUT1 is it has a very low Km. And this I have already discussed in my video on enzymes, what is the significance of Km. So I am not going to go into that much detail, but yes, if any enzyme has, anything has low Km, that means it has more affinity. So I can simply say that minimal basal uptake. This I will explain once I finish up with this minimal basal uptake. Okay, so this is GLUT1. Then we come over to GLUT2. Now GLUT2 is located in the beta cells of pancreas. Very, very important, because they have to release insulin, isn't it? Beta cells of pancreas. Then you can say that the sinusoidal cells of liver. Cells of liver. Right. And always remember, if we talk about the function mein they have a high Km. Means they have very low affinity. Okay. Others are also there, but I am not mentioning all. I am just writing mainly it is like beta cells of the pancreas. Okay. Then we come over to GLUT3. Now, if we talk about GLUT3 is specifically for neurons, very, very important, and for the kidneys and placenta. Now, since they are required for the neurons, their funda is the same as they have a low Km. So, just keep this thing in mind, GLUT1 and GLUT3 have low Km. Then we come over to, after this, we will come over to GLUT4. Now, GLUT4 is different. Why? Because GLUT4 is the only insulin-dependent transporter. So, I can simply write GLUT4 is insulin-dependent, and the most important feature which distinguishes it from the rest of the transporters. And it is responsible for the transport of glucose to 3 main areas. Mnemonic is has, just to remember it. So, here H stands for heart muscle, A stands for adipose tissue, and S stands for skeletal muscle. And obviously insulin dependent. I am writing it here also because this is the most important property of GLUT4. And this is frequently asked in the exam also, that which of the glucose transporters is required for its transport to the myocytes. So always remember, to the skeletal muscle, to the adipose tissue, to the heart muscle, GLUT4 is required. Okay. Then we come over to GLUT5. After GLUT4 comes GLUT5. Now always remember, GLUT5 is the only transporter for fructose. Again, this is facilitative diffusion, and if we talk about the location, the most important one is because fructose is required to give energy to the sperms. So it is like in the testes and sperms, obviously. Okay, and then the intestine. The most important point is you have to remember the sperms, and GLUT5 is facilitative mode of transport of fructose. Then we come over to GLUT6, GLUT7, GLUT8 and GLUT9. Okay, the most important ones I have discussed, these are also important, but again the most important one here is, suppose if we talk about GLUT6, GLUT-C is basically present in the spleen and leukocytes. And if we talk about the function, it's a pseudogene. And if we talk about GLUT7, very important, it is present in the smooth endoplasmic reticulum. And if we talk about smooth endoplasmic reticulum, it is required for the transport of glucose 6-phosphate, because glucose 6-phosphate needs to be converted to glucose in the presence of enzyme glucose 6-phosphatase, which is actually present on the border of endoplasmic reticulum. And this is a very important transporter, because when we are going to study gluconeogenesis, when we are going to study glycogenolysis, this will be the main enzyme, because ultimately whether it is happening in the liver, whether it is happening in the muscle, glucose 6-phosphate needs to be converted into glucose, क्योंकि body को किसकी जरूरत है? Body requires glucose. So glucose 6-phosphate is converted into glucose by glucose 6-phosphatase, and for that it has to come to the endoplasmic reticulum, and for that GLUT7 is the one which is required. GLUT8, the blastocysts, right? As such, no function. Now coming over to GLUT9. GLUT9 is again very important, and GLUT9 is basically the transporter of uric acid, and it is present in the intestines and the kidneys, and it is required for uric acid or urate transporter. And if there is a defect in this, obviously the defect will lead to gout. Okay, so these are the different type of glucose transporters. Now the most important one, I will just highlight, GLUT1 has low Km. Similarly, it is for RBCs. GLUT3 is for neurons. Again, it has a low Km. GLUT4 is insulin-dependent. So it is for heart muscle, adipose tissue and skeletal muscle. That is why we always advise a diabetic patient to go for exercises. Why? Because it will obviously increase the uptake of glucose, and then automatically the level of glucose will come down. Okay. Then GLUT5 is for fructose, and GLUT9 is for urate, or you can say defect leads to primary gout. And now the question is, if I have mentioned GLUT1 is for RBCs, GLUT3 is for neurons, it has low K, which means it has high affinity. And if I have to explain high affinity in a simplest language, I can simply take the example of like BFFs, best friends forever. Best friends forever means even if a person is not saying anything, the other person will come to know that the person wants this thing. So it's like RBCs and the neurons have such a kind of affinity with glucose that even if the concentration of glucose is say 0.00001, milligram per dl also, they automatically will give energy to RBCs and to the brain. This is very important. And this type of terminology, if we talk in scientific language, we call it as minimal basal uptake. Minimal means least. Basal is again the least, when the body is like, totally in dearth of that uptake. So this one has to keep in mind that GLUT1 and GLUT3, no matter what is the status of the body, they will always work. And even if the concentration of glucose is barely, very, very less, then also they will require, because RBCs, again, blood has to be pumped from head to toe, obviously, so it requires energy. And neurons, brain, because brain has to be active. If for a second also, if brain does not get oxygen, automatically no energy will be given. Right, so these are the two important points. And these are the glucose transporters. Now the question is, we have discussed sodium dependent, sodium independent, insulin dependent, insulin independent. Now the question is, what we get is, we actually get glucose, fructose and galactose. All the transporters, and say this is your cell membrane. And just now I told you that it is bidirectional. Now the question is, glucose will also enter, fructose will enter, galactose will enter. Since it is bidirectional and we know that glucose, fructose and galactose are soluble in water. Okay, carbohydrates, they are soluble in water. The moment they enter inside the cell, what is the guarantee that they are going to stay inside the cell? They might go back in circulation. Nobody can stop them, isn't it? So what happens is, the moment they rest on the cell membrane, immediately a charge is put on them. So kinases, it's like, kinases is ready to welcome glucose, galactose and fructose. Kinases, which we have discussed in my video on enzymes. I told you kinases will always attach phosphate, if you people remember, and if you don't remember, you can just have a look on that video. So kinases attaches phosphate, and it is very interesting also, the moment glucose enters, fructose enters, galactose enters, immediately phosphate is attached. Phosphate kya hai? It is negatively charged, PO4 2 negative. Once the molecules are with a charge, can they go back? No. So if they are charged, I would say glucose, fructose and galactose, if they get charged, they cannot go back. Right, so this is the most important feature of the entry or acceptance of the principally physiologically important monosaccharides inside the cell. So the first step, no matter what happens, wherever they are going, they need to be phosphorylated first. And I have told you the reason why phosphorylation is required, because cell membrane, this is the most important property of cell membrane, that it is selectively permeable. Selectively means it will allow only selective molecules to pass through it. The moment it gets a charge, it will not go back. Because if glucose, fructose, galactose in a normal individual starts going back, first of all, there is no concept of biochemistry. There is no question of the reactions. And second is, the osmolality of the entire blood will be automatically hampered, because there will be increased concentration, all of them in blood, not inside the cell. Okay, so the entire science will only fail. The medicine will also fail. Okay, so always remember these physiologically important monosaccharides, the moment they enter inside the cell membrane, they need to be phosphorylated first. And phosphorylation ke liye, the only enzyme which is present, the only class of enzymes which is present is kinases, which belongs to the transferases group or the class 2 of enzymes. Now what happens is, they will be phosphorylated. So if I talk about glucose, glucose will be phosphorylated by hexokinase and glucokinase. Both are the main enzymes, glucokinase. I will tell you what is the difference also when we will study the metabolism. So you can say hexokinase and glucokinase are going to form glucose 6-phosphate. Charge. If I talk about galactose, galactose will undergo phosphorylation. Everywhere you will see kinase is added as a suffix. So it will be galactokinase. Obviously, galactose 1-phosphate instead of galactose 6-phosphate. Why? Because again, galactokinase is a specific enzyme which phosphorylates only at first position. And if we talk about fructose, again the enzyme is fructokinase. And it is going to form fructose 1-phosphate. So now, these are the ones, these are the substrates for oxidation or for different reactions to give energy to the body. So this is how, first of all, digestion and absorption of carbohydrates starts. And always remember guys, the digestion of carbohydrates always starts from the mouth. Proteins ka never starts from the mouth because of the presence of salivary amylase, okay, in the mouth. So these carbohydrates are digested like this, and proteins ka digestion, it starts from the stomach. We know that pepsin is there which works at a very acidic pH. Now the question is, all of them have entered. What is the fate of glucose 1-phosphate or glucose 6-phosphate, galactose 1-phosphate, fructose 1-phosphate? So this will lead to the metabolism, which is actually the crux of biochemistry. Now, first of all, what is this metabolism all about? You have studied in your previous classes also. Metabolism basically kya hota hai? It has two classes. One is anabolism. Basically, it is of two types, I would say. Anabolism, and second is catabolism. Up, if we talk about ana means synthesis of something. So obviously, if something is synthesized, energy is given. So I can say that if A plus B is synthesizing C, it requires energy. Catabolism is breakdown of something. That means C, whenever the body requires energy, C will be broken down to A plus B. So energy will be liberated. So anabolism plus catabolism is metabolism. So whenever the body requires energy, because we are talking, we are walking, we are writing something, we are doing so many metabolic activities. So whenever the body requires energy, the product is broken down, and whenever the body is at rest, we are eating something, that thing will be conserved to produce a product. But the net flux of energy will remain the same. That whatever energy is utilized. Now pay attention here, this is very, very important. Whenever energy is given to synthesize something, that energy will be liberated during the degradation of that particular thing. Or I can say that energy can neither be created nor destroyed. It can be simply converted from one form to another, and that is the basis of first law of thermodynamics, which we have been studying since, donkey's years. That energy can neither be created nor destroyed. It is simply converted from one form to another. So what is happening is anabolism and catabolism are going hand in hand, so that overall they will form metabolism. Now, since this is occurring in the body, because there is a difference between in vivo and vitro. Okay, so whatever is happening inside the body, so we can say that our body is actually undergoing 3 different stages, 3 or I can say 4 different stages. Just suppose first thing, when we get up in the morning, when we are having our first meal, that is breakfast. Okay, after breakfast, we will have our lunch. So we are giving a time interval of 3 to 4 hours. Suppose if we don't like the lunch, we can skip lunch. If we don't like the menu in the lunch, if we are skipping lunch, that means we are increasing our time duration, and in between we don't get any time to eat. So suppose if we had a first meal say around 9 o'clock, we are skipping the lunch, and suppose we wish to have dinner say after 12 hours. So what is happening to the body? The body is undergoing that fasting wala clock. So from 9 o'clock in the morning till 9 o'clock at night, since we didn't have anything, aisa to nahi hai we will die. We are not going to die. We will perform all the activities which we have to perform. So how the body is meeting the demands, the I would say the status of the body. That is what is the reactions all about. Okay, and suppose let's put it that way, ki we had breakfast, we don't like the menu in the lunch, we don't like the menu in the dinner for that matter also, we sleep like that only. We get up in the morning. No doubt we will have a little bit of kind of faintish feeling, but yes, we won't die. Okay, so what is happening? We are increasing again the time interval of fasting. So whatever reserves are there, then liver will become the most active organ because it has to supply energy. It has to supply energy in the form of food. So RBCs also will be functioning because blood has to function, heart will also function and brain will also function. Okay, so that is what is. So we always divide this interval into different stages. So always remember the most important stage which we say is it is the fed state. Fed state, well-fed state means when we had our first meal. So fed state ka I can simply say that if we talk about the duration, it is post, or you can say, yeah, I'll just make it more simple, food intake. When we are taking in food. Ab, when the fed state is there, whether we are having carbohydrate, whether we are having protein, whether we are having fats, whatever we are having. So in the fed state, everything will try to go and form that form which will be stored in the body. Just say if I talk about glucose, glucose automatically will undergo oxidation to give energy. This is so there. But glucose will be conserved to form fats. And fats will be stored in the adipocytes. Okay. Ab now see, suppose after food intake, when the body undergoes post-absorptive stage, that becomes the fasting stage. So fasting stage we can say it is going to last for say 4 to 16 hours. Right? Then late fasting will come. If I talk about late fasting, it will be say 16 to 48 hours. Prolonged fasting is known as starvation. So if we talk about prolonged fasting, it will be say from 48 hours to say 3 to 4 days. I will write 4 days. 4 दिन तक हम कुछ नहीं खाते, then the body will undergo prolonged starvation. So you can see that there is a difference in all the stages. So it will be prolonged starvation. Okay. Now the question is that how the body meet the demands like that. So if we talk about fasting stage, which is like some 4 to 16 hours, the only source of energy is glycogenolysis, and that too in the liver. So hepatic glycogenolysis. Right. Late fasting ho gaya, the time interval is increasing. It is gluconeogenesis. All the processes we are going to discuss in detail, but before starting up with the metabolism, we should have a sound knowledge of this. Being a doctor, one should understand what the patient is undergoing through, whether it is a fed state, fasting state, and what are the repercussions of that also. Prolonged fasting may what happens is the fats will be degraded. And you know the fats are degraded to form acetyl coenzyme A, which form ketone bodies. And ketone bodies clinically very, very important. In a person who is suffering from diabetes, if he is not taking proper meals, not under proper medication, there are most likely chances of that patient to undergo diabetic ketoacidosis. Diabetic ketoacidosis is the worst state in a diabetic individual, when the body will start giving the smell of that nail polish remover, that acetone kind of removal. Okay, because he has undergone fasting, because obviously diabetes में क्या हो रहा है? Glucose तो है, the concentration of glucose is more, but it is not reaching the cells. So it is equivalent to starvation only, right? And if we talk about prolonged starvation में actually the period is after 4 days, 4 to 5 days. And the last source of energy, when the body has given up everything, the last source of energy is the protein. So there will be muscle proteolysis. the entire body will undergo putrefaction, and that is why it starts giving that foul smell also, because there is breakdown of the proteins. So to understand this is very, very important. First of all, which is also known as the fed state. Fed state mein all the anabolic reactions will occur. Okay, glucose to fats, this is anabolism. But if we talk about fasting, late fasting, prolonged fasting or prolonged starvation, the body has to undergo degradation, giving energy to the body. So if we talk about 4 to 16 hours, glycogenolysis, 16 to 48 hours, gluconeogenesis, 48 to 4 days, ketone bodies, and after 4 days, muscle proteolysis. So this is actually the metabolism revolves around these things. Now, after this, then we should know that what are the different hormones which are acting in the well-fed state or in the fasting stage. So obviously, I can say that, or overall if I have to say that, I can simply say that in the fed state, generally the anabolic reactions will occur. Anabolic reactions. And in general, we say all the anabolic reactions occur in the cytoplasm. So I can say anabolic reactions in cytoplasm, but exception is glycolysis. Glycolysis, even though it is a catabolic process, but it occurs in the cytoplasm. So in general, they occur whether it is synthesis of fats, whether it is synthesis of cholesterol. So fed state mein anabolic reactions will occur, which occur in the cytoplasm. Exception is glycolysis. And if we talk about the breakdown of something, breakdown will occur in the fasting stage, obviously fasting or starvation. Okay. So in the fasting state, all the catabolic reactions will occur. Catabolic reactions. And we know catabolic oxidant is required. So simply in the mitochondria. Right? And here exception is gluconeogenesis. So this one has to remember, one is the fed state, other is the fasting stage. So always remember all the anabolic reactions occur in the fed state, exception is glycolysis, and all the catabolic reactions occur in the fasting stage except gluconeogenesis, which is actually an anabolic process, but just now I told you it occurs in fasting stage. Okay, now here since we are talking about the metabolism of carbohydrates, so we have to remember one thing, that these carbs will undergo metabolism again in two stages. One class will include the major pathways and the other will include the minor or the alternative pathways. The difference is alternative pathways will be only in the selective organs and whenever they are required, and major means all the time they will be working. So if we talk about the major pathways, the first one is glycolysis, TCA cycle, glycogenesis, glycogenolysis, and gluconeogenesis. These are the major pathways. And if we talk about the minor pathways or alternative, one is hexose monophosphate shunt. Commonly we call it as HMP, shunt. But it is always known as a shunt. I will tell you the difference. What is the difference between shunt and pathway? Second is uronic acid pathway. And the other minor pathways are galactose metabolism and fructose metabolism. So this is the list of major pathways, this is the list of minor pathways. And if we talk about glycolysis, glycolysis is strictly catabolic pathway. This also you have to remember, very, very important. TCA is the only amphibolic pathway. Why? Because it is occurring in both. It has anabolic as well as catabolic role. Glycogenesis is anabolic.
Dr. Pooja, battery is below 50.
Glycogenolysis is catabolic and gluconeogenesis is anabolic. So this is how we are going to study the major pathways and the minor pathways, and whatever metabolism is there of the carbohydrates, it is hidden inside this. So in this video I will just stop here, and if I have to just conclude everything, the most important point which you have to remember here is that there are sodium dependent transporters, sodium independent transporters. So I can say the most important points which you have to focus on this is that sodium dependent transport, whether it is 1, whether it is 2, it is known as secondary co-transport. First point. Second point, we should know that GLUT1 is exclusively for RBCs and GLUT3 is for neurons, and they have low Km and high affinity for glucose. And third important point is that GLUT4 is insulin dependent, and it is required for the transport in the cardiac muscle, in the adipose tissue, and in the skeletal muscle. So these are the important glucose transporters. So here I finish up with the video on glucose transporters. In the next video we will be taking up the major pathways, the minor pathways in detail. Thank you so much.